let's do 9.2. This one's all about the sugar, and that sugar, something I know you all like. Last 9.1 is about the xylem, about water. 9.2 is all about the phloem, which is where how plants move sugar around in their structures. And we're still looking at this structure and function idea. And the main thing, we have, tubes, these phloem vessels that we talk about. They're basically these kind of fancy tubes that help transport the sugar. So the function, the structure, which is these tube shapes, helps with the function, which is to transport sugar. So remember, look at anything in biology, chemistry, physics, the structure of what we're looking at has a lot to do with the function. And remember, like enzymes, right? Enzymes have their active site, and that structure of that active site on that enzyme relates to what their function is, what substrate they will join to. You all remember the hydrogen peroxide and catalase in the potato lab. One thing that we'll get to at the end of 9.2 is this really cool experiment where they took aphids and put radioactive elements in their isotopes and were able to trace how long it took for sugar to move along the plant. So again, radioactively labeled carbon dioxide, radioactively labeled sugar. Just using this radioactive labeling really helped. This one development in science really helped to elucidate, explain a lot of the phenomena scientists have been seeing for a long time. But once they had those radioisotopes in the 1930s, I believe, it was ready to go, ready to use that for elucidating, figuring out all these complex pathways. So yes, the single development in science gave rise to dozens and dozens of experiments that helped to explain the world around us. Biology, since we're focusing in on biology. So here we go. Xylem, which is what we talked about last time. Transportation of water. And remember, always going up. Up here are the leaves. Down here are the roots. And as you'll see, phloem, again, a tube-shaped structure. It is by and large going in the opposite direction because you have the sugars being created in the leaves. So up here, the source cell. We talk about phloem, we're talking about source and sinks. So source is where it starts, where it's made. Sinks are where it ends up. So a source equals the leaf, and with the sink, probably the roots or the fruit, someplace where the plant is storing that sugar. And you all hopefully enjoy fruits, and that is a sink of all that sugar that's being produced in the leaves. Transportation, we call it translocation because let me rewrite that. Translocation. Of sucrose. And this is using energy, pumping the sugar into these phloem cells and moving it. And you can kind of see, you can see higher concentration up here and then lower concentration down there in the sinks. So water is not compressed, and water does not compress. You can't squeeze it like you can a gas. Solids also are incompressible. But gases, as you know, we can compress gases. So sucrose, you'll see this blue here is looking at water, H2O. And the sucrose and the water get loaded into the phloem. And you see, because there's a high concentration, let me get better contrast. So we have a high concentration of the solute, which is the sugar. That's pulling water in. Remember, water moves from high water concentration to low water concentration. Or if you're looking at solutes, water wants to rush to that high concentration of sugar or salt. Those are typically the two ions that we're addressing in biology. And then you see the sucrose coming back out at the sink cell, in the storage root, like a carrot or a potato. That energy is being stored for a later date. Take up water by osmosis. Oh, turn that in. Fill this out. So that's what we talked about here. And positive pressure, we see high pressure of water. All that water is being sucked in or moved in from osmosis. And to relieve that pressure, these substances move down to those sink cells. And this recycles water. That is water from the sink to the source. Hopefully that's somewhat legible. So active transport. Remember, active transport. You see active, you know you're using ATP. Getting that sugar into those phloem cells at the source. Here we go. You see the protein pump there, using your ATP and pumping that hydrogen. And as the hydrogen moves in, so does the sugar. So the sap, you've all heard of maple syrup. Maple syrup, that sap, and it's very sweet and sugary. And that's dissolved in the water. And sucrose is the most prevalent, and it's good because plants, they break down glucose. They got to break the sucrose because that's two glucose molecules, or it's a fructose and a glucose, if I remember. Snaps it in half, breaks it in half. Then it uses the glucose for energy and the fructose. But sucrose, not usually. By being transported as sucrose, it means the cells that it hits along the way is not going to use it up. So it's not used for respiration. Oh, and by the way, there's also minerals. Let me add that. So sugars and minerals. Right there. There we go. So sugars, minerals, a bunch of important molecules moving down through that phloem to make sure that plant gets the energy and the stuff it needs to run all of its processes. Here's another model. You've been seeing this diagram a lot, called the pressure flow. And what I explained earlier, if you have all the sugar in here, it's going to bring water in, and it's going to build up that pressure. So build up of sugars in the phloem cells become they're hypertonic, because that sugar is being added in there, and in hypertonic environments, the water is going to move towards hypertonic. It's like the onion cell lab, the red onion cell lab. We put salt water around our red onion cells. Water is going to get sucked out. It's going to go towards the hypertonic solution. So let me clarify, add that. Water moves toward hypertonic. Hypertonic solutions. Because it's hypertonic, because you have a whole bunch of solute there, it's going to bring water in from the xylem via osmosis, which doesn't take any energy, and that's going to increase your turgor pressure, and that kind of pumps up. Turgor pressure is like if you blow up a balloon, it stops being floppy and starts getting firm. So the phloem now, because we have that high pressure in the source area in that phloem, we're now going to be translocating that phloem with the sugars down towards the sink. So we load. So loading sucrose and some amino acids. From the source, which are the leaves, right? Or the stems. Some plants have some photosynthetic capabilities on their stems. Wherever you see the green, you say, oh, there's chlorophyll there. Photosynthesis can occur. There's also, we'll get into this later, a cool little cell called a companion cell that helps to sustain these phloem cells. These phloem cells are like race cars, stripped down. They only do one thing, just having those side walls sustained, so they can hold that sugar. And these companion cells, they have more things like mitochondria, ER, lysosomes, and can sustain. The same thing happened, if you remember back in reproductive. Sperm cells are stripped down, very small. Remember, so they can move great distances, but that means they don't have a lot of the cellular machinery they need. Well, they have companion cells, Leydig cells, that help to keep the sperm alive. So this mass flow, we have one thing we'll get to later. There's these things called sieve plates, basically cell walls that are kind of self-eroded. These holes made in so that the fluid can move through. So crossing that sieve plate all the way down to the sink or wherever this is being stored. So that's called unloading. At the sink/storage, wherever it's being stored. Like I said, it could be like in the roots somehow. Roots. Remember, if it's a fruiting plant, it'll put a lot of this sugar energy into the fruits. Yeah. And then water is released at that point and then goes back into the xylem. And it's just this really kind of cool back and forth. The water going up, coming down, going up, coming down. The sugar is only going one way though. That sugar, most of the time, I know there's exceptions to that. I can't think of one, but I know there's exceptions, like any rule, right? There's an exception to that. And here to finish up translocation theory, the sap has to go across the sieve plate. It's the same as the last one. Hold on. Yeah, I forgot to separate those two, but I'll show you anyway. There's still like I mentioned before, along here, there's not much cytoplasm content. Not much in there. So it doesn't have a lot of the organelles. But those companion cells, they're actively, they unload the organic molecules into the phloem, and that requires ATP for this companion cell to kind of move sugars in there, to move sucrose in from here to here. Takes energy. Oops. Oh, there might be some other issues I looked at with what slides you see in your packet and what slides I'm going over here. Just focus on the slides I have here. If I think I had some repeat slides from 9.1 and 9.2, so just ignore those, by the way. And they're stored, sucrose usually is stored as starch at the sink, and then that water is recycled.